Thanks for joining us for today's webinar, Using Genomics and Genetics to Unravel Virulence and Secondary Metabolism in Cercospora, a lesson from Cercospora ZA Matus and C. beticola, presented by Dr. Sandeep Sharma Khatiwada. Sandeep earned his Ph.D. in Cell and Molecular Biology from the University of Arkansas in Fayetteville, before joining Cornell University in 2018 as a postdoc researcher in plant pathology with Dr. Sarah Pethybridge. His current research involves developing novel strategies to mitigate the production of cercosporin, a phytotoxin, and other harmful secondary metabolites from different filamentous fungi. With that, I'll hand everything over to Dr. Sharma to begin his presentation.
Thank you very much for the introduction, Caitlin. And I would also like to thank GeneWiz for giving me this opportunity to share my work with a broad audience. And I'm excited because this has been a slightly different experience for me in presenting in a webinar format. So today I'm going to give a talk about some of the work that I have been doing since my PhD and my current work as a postdoctoral associate. I'll be talking about genomics and genetics to unravel virulence and secondary metabolism in Cercospora. Specifically, I'll be talking about my work with Cercospora ZA Matus and Cercospora beticola. Before I go into my research, I'll like to give a brief introduction about myself. So I am a native of Nepal, and I did my BS in agriculture from Assam Agricultural University in India, after which I did my MS in agriculture, specializing in molecular biology and biotechnology from GB Pant University of Agriculture and Technology, also in India, after which I did my PhD in cell and molecular biology from the University of Arkansas. And I'm also active on Twitter. You can follow me, which you can see on the slide. But my broad interests are in molecular plant-fungal interactions. I'm specifically interested in Cercospora species, which I worked extensively during my PhD and currently in Cornell University. So you may think, why Cercospora species? And which brings us to the next slide. Because Cercospora, they cause diseases in numerous crops. We have some examples here is Cercospora beticola, which causes diseases, leaf spot diseases in beets, spinach. Also have Cercospora ZA Matus, which I mentioned, and Cercospora zeana. They cause gray leaf spot of corn. We have other Cercospora which cause disease in soybeans, specifically Cercospora sagina and Cercospora kikuchii. And we have in mustard Cercospora brassicicola and Cercospora jansiana in rice. Now Cercospora, they belong to class Dothidiomycete in a big phylum Ascomycota, which is one of the biggest phylum in filamentous fungi. Cercospora are basically foliar pathogens, so they mostly infect the plant via leaf. We still don't know if they infect through stem or roots yet. They cause infection by entering the plant through stomata. And so far what we know in Cercospora is they overwinter in plant debris, and seeds have also acted as a potential source of inoculum. However, how they reach the seeds, how they infect the plant and are able to colonize the seed is not known yet. Beside overwintering in plant debris or entering the field through seeds, they are also known to infect numerous weed plants, which include wild mustard, dockweed, lamb's quarters, safflower, just to name a few. And they are primarily controlled with fungicides. Just to give you a scale of how devastating Cercospora field is, you can see different field scenarios here. You have a cornfield on top left, and in the middle you have a beet field from New York. Bottom left, you can see a cornfield that's completely devastated with Cercospora ZA Matus. That field is from China. And bottom in the middle, you can see a lesion on rice plant that's caused by Cercospora jansiana. And on the right, it's typical leaf that's infected with Cercospora sagina, causing the disease frog eye leaf spot. Now, like I said, that previously said that Cercospora were primarily controlled with fungicides, and that's being a big issue right now. Is fungicide resistance is widespread within populations of Cercospora. So there isn't a complete resistance to most of the Cercospora species in crops other than Cercospora sogina, where we have a major resistance gene being discovered. However, there is development of races against those R genes, and so we feel that that's not a viable strategy for managing. So the fungicides that are commonly used in agriculture against Cercospora belong to FRAC groups 3, 7, 9, 11, and 12. The FRAC is Fungicide Resistance Action Committee. However, there is widespread resistance to FRAC groups 11 and FRAC groups 3 that are currently being deployed in field situations. Hence, I feel that alternate management strategies are essential to control Cercospora. Now, this brings me to my research is utilizing Cercospora beticola and Zeomedes to develop alternate management strategies. Before going to Cercospora beticola and Zeomedes, just giving a brief background on these two systems. So Cercospora beticola causes Cercospora leaf spot of table beet, sugar beet, and Swiss chard, which fall under broad Beta vulgaris, subspecies vulgaris. And recently it's also been reported on spinach, and it's widespread in USA and Europe. On the other hand, Cercospora zeomedes causes gray leaf spot of corn, and it's widely spread in corn growing areas of USA, specifically in the Mississippi Delta region and China. Now, the interesting thing about Cercospora zeomedes is there is a sister species called Cercospora zeana, which is more predominant in South Africa and Eastern USA, like the New York, Pennsylvania, New Jersey, etc. They are both Cercospora zeomedes and Cercospora zeana cause gray leaf spot, but they are two different species. Now, why am I focusing on Cercospora beticola and Cercospora zeomedes is they're excellent systems for genetic studies, which will, cover the whole of Cercospora. Their genomes has been sequenced and assembled. The Cercospora beticola genome, the isolate that I'm working on, TB14085, was sequenced in the lab I'm currently working in. It's called Evade Lab. And the genome is about 35 MB with 97% complete, and it's publicly available in PubMed. Cercospora zeomedes, the isolate SCOH1-5, was sequenced by my previous lab through JGI, and it's a slightly bigger genome, 46 MB, and the genes are about 12,000 genes, has been annotated. Biggest advantage is also both these species, both these isolates are routinely amenable to transformation. We've tried both agrobacterium-mediated transformation and PEG-mediated transformation of protoplasts. However, most of the genetic studies focused on these two Cercospora are pathogenicity and secondary metabolism. In my previous lab, working with Dr. Bertblum, CRP1 was the role of CRP1 was dissected in Cercospora zeomedes, and in Cercospora beticola, most of the genetic studies focusing on virulence is on the genes ADE1 and Cercospora beticola NIP1. Also, Melvin Bolton's group in NDSU focuses a lot on cercosporin cluster in Cercospora species, specifically Cercospora beticola, which I'm going to go into introducing you to what is cercosporin. Cercosporin is a secondary metabolism that's well reported to be produced by different species of Cercospora. It is one of the numerous secondary metabolites produced, and it's been known that it's produced by all Cercospora species. It's broadly a non-host specific toxin. That means it's not limited by a host. whether it's a maize pathogen or a rice pathogen or a soybean pathogen, like everyone produces this. And it's photoactivated. And as you can see in the figure, cercosporin needs light to be produced and activated. And cercosporin breaks down to produce reactive oxygen species, which go and damage the membrane of the plants, which causes cell death. And this causes the plant contents to be leaked outside, which in turn can be utilized by the fungus to absorb the nutrients from the plants. And going on to in depth about cercosporin in Cercospora species, what's being known is cercosporin is produced by this cluster of genes, which is called cercosporin cluster. Initially, there were 8 genes from CTB1 through CTB8. It's a polyketide synthase backbone is the main gene CTB1, and it's a group of transferase and transporter oxidoreductase that help to produce this molecule whose structure you can see on the right. However, recently, the work from Melvin Bolton's group in NDSU expanded this cluster to add 4 more genes, which has been named as CTB9 through CTB12. Not only they have expanded the cluster, but they have also identified that this cluster is not just present in Cercospora species, but is also present in Coletotricum species. So there is evidence of horizontal cluster transfer from one species to another. On the left, you can see 2 plates. On the top is a PD plate with Cercospora, but the plate is colorless. In the bottom is bright red pigmentation. That pigmentation is due to production of cercosporin. So if you expose that plate to constant light, the fungus produces cercosporin. So it's not just in planta that cercosporin is being produced, but it can be also produced in artificial medium. Although we know what cluster is producing cercosporin, what we don't know is the genetic regulation of cercosporin itself. So far, what we know on the regulation of cercosporin is it is influenced by pH, it is influenced by light, and it is influenced by nutrition. However, what are the genetic factors, like what genes are responsible for regulating this with this cluster within Cercospora was largely unknown. So in order to dissect the regulation of cercosporin, I decided to deploy forward genetic screens. Now, going to forward genetic screens in filamentous fungi, it is powerful for gene discovery and function. It was mostly used in model systems such as Saccharomyces cerevisiae and Cryptococcus neoformans. Recently, the deployment of forward genetic screens has been accelerated due to availability of different tools such as transformation systems, different gene knockout strategies in filamentous fungi, phenotype screenings in a large scale and high throughput. And so it has been widely used in fungal pathogens of plants to look at genes regulating virulence. Some examples include Magnaporthia rhizae causing rice blast in rice, Fusarium verticillioides, Phomopsis longicola, and also Botrytis cinerea. Coming to utilizing forward genetic screens to dissect cercosporin production in Zea maydis. So this was my big project during my PhD, and I wanted to use this to see initially or identify genes that were potentially regulating cercosporin production. So basically, coming to a forward genetic screen, we used 2 different transformation techniques. One was protoplast mediated transformation, or PEG mediated transformation of protoplast. And in this, we used a GFP reporter with geneticin resistant selection was used. And the second one I used was agrobacterium-mediated transformation. And the plasmid I used was PBHT2S-GFP, which I'll talk in details in a moment. In this, the resistance marker was hygromycin and also the green fluorescent protein. So basically, in both of the techniques, we had two different selection markers. One was the fluorescent marker and one was the antibiotic marker. And on the right, we can see like how we go about, only selecting those colonies that were growing on the plate containing the antibiotics, but also fluorescing green. But the colonies that were not fluorescing were not selected for future analysis. So I'm going to give more details about the plasmid that I used in agrobacterium, because this method was I used more extensively rather than protoplast mediated. And so you can see on the screen, I have the plasmid map showing the GFP, the promoter, and the hygromycin phosphotransferase, the antibiotic selection, which was present within the right and the left border of the plasmid. This plasmid I used in the agrobacterium strain AGL1, which was first used by Lazo et al. In 1991. This PBHT2S GFP is modified from PBHT2, originally used by Mullins et al. And the plasmid I have also sequenced and assembled, so I know the sequence of the plasmid. And we had routinely used this plasmid to transform, Fusarium verticilioides for Mopsis longicola in the lab. So in my screen, I generated more than 3,000 mutant strains of Cercospora zeomatis and screened for cercosporin production. And just like how I showed you previously how cercosporin turns red when it's exposed to light and you inoculate Cercospora in PDA. So that's how my screening was done. So in the left, you can see the colonies 7 days after inoculation, and on the right, you can see the colonies 10 days after inoculation. So in the center is the wild type isolate, the SCOH1-5 of Cercospora zeomatis, and surrounding are some of the mutants that are displaying different cercosporin phenotype. So the two colonies on the top you can see are like really bright red, which show that those two isolates are probably secreting and producing high amount of cercosporin compared to the wild type isolate in the center. And in the bottom, you can see 3 other different fungal strains that either are not producing cercosporin or are producing less cercosporin compared to the wild type isolate. And not just cercosporin, there was one particular mutant which actually stood out in the screen, and this particular mutant that I have labeled A was like really white in colony morphology. There was no, so on the B, you can see it's the wild type, the SCOH on V8 medium, and the A is like white, and also in dark, it seems to be producing this really bright red pigmentation on V8. Initially, during our screen, we didn't know what it was. It was just fascinating to see like a completely different strain, which looked, morphologically different than the wild type isolate. So after the screening was done, a big issue is how to identify the mutation in forward genetic screens. And this identification, it represents a big bottleneck, because you have these, large collections of your mutant strains, and you have done your screen, and now, you could have hundreds of interesting candidates that you would like to identify the mutation. But it's a big problem in, even handling a few strains. Now, the typically used methods were genome walker, tail PCR, 5' race, plasmid rescue. Although I'm saying these are typically used methods and they were previously used, but they were really cumbersome, and you needed a lot of sequencing to be done, and you could spend many days doing genome walker before you could identify mutation in a single isolate. However, with the advent of genome technologies and reduction in prices of whole genome sequencing, people started actually using whole genome sequencing to identify where the insertion of the mutagenesis cassette was. We developed a restriction enzyme-based method to identify. We named it MonsterSeq. It's been published. However, today what I'm going to be talking about is target enrichment sequencing, which can be efficiently used to sequence many samples and also correctly identify where the mutagenesis happened. Because in both agrobacterium-mediated transformation and in protoplast-mediated transformation, we have a mutagenesis cassette that goes and randomly inserts itself in the genome, but we don't know where. So this target enrichment sequencing is basically what is a form of kind of exome sequencing that they do in humans. However, its use in fungal genetics is still limited. And what it basically does, it enriches or sequence only selective regions of the genome, and hence it's high throughput, and you can sequence multiple samples simultaneously. Also, you don't necessarily need a whole genome sequence information, and not just that, you can also identify single and multiple insertions at the same time, which is a big advantage over the existing methods such as genome walker or tail PCR. Now, how do you go about doing this target enrichment sequencing? So in the figure, let's say you have this mutagenesis cassette, which is in white, which goes and randomly inserts in the genomic DNA somewhere. Just to give you a heads up, we know the sequence of this mutagenesis cassette, but we don't know the sequence of the genome or the genomic DNA where it's inserted. So what we did was, towards the border of the mutagenesis cassette, spanning over 240 base pairs, we designed biotinylated probes. And as you can see in B, when you prepare your sequencing library, you are fragmenting your genome of the strain into small fragments, let's say 300 base pairs or 150 base pairs or 400 base pairs. And then you use these biotinylated probes to pull down or enrich only those reads that have part of your mutagenesis cassette in white and also have part of the genome. And you don't pull down those regions in your sequencing libraries that don't have part of your mutagenesis cassette. That is rest of the genome of the fungus, which is not going to give us any information about the insertion. And then after you enrich those reads that have part of your mutagenesis cassette and part of the genome, I used the Ion Torrent platform to sequence these reads. And when you map these reads onto the genome, it'll specifically let you know where your mutagenesis cassette inserted during your transformation event. In my screen, when I told you that I transformed and screened more than 3,000 mutant strains for their ability to produce cercosporin, I was able to identify about 80 mutants that were really interesting. Like I said, some of the mutants were overproducing cercosporin, some of the mutants were producing less cercosporin compared to the wild type strain, and some mutants did not produce any cercosporin. So using the target enrichment sequencing method, I was able to, in the collection of these 80 interesting mutants, I was able to see that some of the mutants had single insertional events, some of the mutants had double insertional events. I was even able to identify triple and quadruple insertional events happening in my genome. And identifying the single, triple, and quadruple insertional events during your transformation was not possible due to previously used methods of specifically like genome walker. And so this was like really interesting. Like in the same run, you can get different events. Also from my sequencing, I was able to get symmetric and asymmetric insertional events. So what exactly is symmetric and asymmetric insertional events is, so on the left, you can see an asymmetric insertional event where I was able to identify only one side of my insertional cassette. Whereas in symmetric insertional events, I was able to map and identify both the side of my mutagenesis cassette. And the symmetric insertional event was able to directly point out, like, here is the point where the mutagenesis cassette inserted, and the reads that we sequenced would go and map at that exact point. Now, other than identifying, what type of insertion happened, we were also differentiate between, PEG-mediated transformation and agro-mediated transformation when they were sequenced simultaneously. So we can see in PEG-mediated transformation, we had more single insertional events than multiple insertional events. However, we were able to identify in our agrobacterium mediated transformation, we had more, or we had equal proportions of single and multiple insertional events happening at the same time. Also talking about symmetric and asymmetric transformation events. In PEG mediated, we saw both, symmetric and asymmetric happening in equal proportions. And also in agrobacterium, we had equal proportions of symmetric and asymmetric transformation events happening. Although the total there says 109, that is taking into account that some strains had multiple insertional events. So even though the number of strains that I sequenced were 80, but the total number of insertional events is 109 because some strains have more than one transformation event. So when we were able to look at the distribution of mutants across the genome in Cercospora zeametis, which you can see in this circular diagram. Just to give a heads up, this is not the whole genome, but we are only showing the chromosomes or the regions where our insertions were identified. So you could see our insertional events were kind of, widespread across the whole genome and not just focused in certain regions of the genome. We can also see insertions happening in repeats. Those are the ones in gray areas, which was not previously identified through normal, techniques such as genome walker. Was very difficult to identify if the insertion happened in repeat. So coming to the genes that were identified in the screen, I actually had made a list of all the 80 genes. I'm pointing it out to this specific gene that's pointed out with the red arrow that I had identified, the specific mutant that I showed you previously that was all white. So that gene that we identified had actually disrupted this gene called skitolone dehydratase. This gene is present in almost all filamentous fungi, and what we discovered later was this gene is responsible for production of melanin in filamentous fungi. So that's why when this gene was knocked out, my strain was not able to produce melanin, and hence it was white rather than dark black of the wild type strain. And my research, the screen was done way back in 2016 and 2017. And recently in 2020, there's this paper which they published, or it's in preprint. They looked at a transcriptome of 2 Cercospora sagina strains that had different levels of virulence. And one of the genes that's specifically mentioned in this research is the skitolone dehydratase, where there is differential expression in 2 different, strains with 2 different virulence. So what it shows was the screen that I had done was only on cercosporin production, but these genes could be highly relevant in virulence in other Cercospora species. So a lot of the genes that I had identified, I categorized them in different groups, and we mostly saw that they were global regulators, histone modifications, transcriptional reprogramming. They were in growth and development, primary metabolism, signal transduction, etc. Now, doing a forward genetic screen, you also need to show that your mutation is directly linked to the phenotype. So I kind of did, or also as a proof of concept, generated some RNAi lines. So some of the mutants that I had identified in my screens, I selected 6 genes that were, non-producers, less cercosporin producers, and overproducers. And some of the genes like CTB1 that we talked about is the polyketide synthase is directly responsible for producing cercosporin. And, I chose a wide different genes with wide different functions. So when we generated some of these RNAi lines, most of our strains or most of the genes, they showed the phenotype that we had seen in our random knockout, such as you can see the CTB1 on the left and the 4612 strain on the top. They are completely devoid of cercosporin production, which we had also seen from our random insertional mutants. And the 9314 and 832, these strains were, overproducing cercosporin when compared to the wild type strain, the SCOH 1-5, that's also being shown in the middle of the screen. Now, from all this study, we were able to put together these pieces of puzzle on how cercosporin is regulated. So we were able to identify a lot of genes that had functions in epigenetic regulation, a lot of genes that could directly impact through signal transduction, primary metabolism, and a lot of genes that had, played a role in stress response, structural components, growth of development. So we were kind of now getting big picture on these genetic regulators of the CTB cluster. However, at that time, I still don't know, so we have these set of genes that are regulating cercosporin, but then how can we utilize the information from cercosporin to actually look at genes that were involved in virulence or the genes that were involved in causing disease in planta? But then I joined a new lab here at Cornell University, and I had a dilemma, is that whether should I utilize the information from the Ph.D. or should I just start all over? So I decided that, I'll be working in a new system that was Cercospora, Beticola, and beet. So I thought I'm going to do a similar study and, start all over again because it's going to be a discovery mode and it was a different pathosystem. So the genetic screen that I did for my PhD looking at cercosporin production, I did a similar study, like a genetic screen in Cercospora beticola to identify genes that were regulating pathogenicity. So on the left, you can see is the Cercospora beticola GFP strain on a leaf surface, and you can see the stomata. On the right are some of the strains that were identified in this screen. So going back, right now, I'm only doing agrobacterium mediated transformation because I feel that's, or personally, I feel it's easier for me rather than PEG mediated transformation of chloroplasts. And how we go about agrobacterium, it's really simple. You take the spore suspension of your fungus and then you grow agrobacterium, AGL1 harboring a vector PBHG2 SGFP. You co-culture them together and then you transfer them to a selection medium containing the antibiotic hygromycin. So then you select the fungal strains that grow on these antibiotic laden plates, and also you shine light on plate and you can select the fungus that are fluorescing and growing on these antibiotic plates. So on the right, you can see the colony of fungus that were actually selected for this screen. You can see they are fluorescing colonies, and that plate actually contains the hygromycin antibiotic, and you can select for downstream analyses. And then I took these strains from transformation and kind of inoculated them onto beet plants and looked at whether they were able to cause disease or not. So most of my screening for virulence was done in mist chamber trials. So what I did was I made a hyphal, like a suspension on tween in 1.5 ml tubes, and they also had probably some spores in that suspension. And each plant was inoculated with multiple isolates. You can see that each leaf I had inoculated with wild type and my strain. So this fungal suspension, this hyphal and spore suspension, was inoculated with a pipette. I inoculated about 2 or 3 microliters on each circle, and this plant was then incubated in a mist chamber at around 23 degrees Celsius, but also containing very high relative humidity, more than 90% relative humidity for at least 4 hours a day. And these plants were screened after 14 to 21 days. And on the right, you can actually see how these leaves are, or how the plant is being screened. So you can see some of the mutants strains are actually producing these lesions, these typical leaf spot lesions. And you can also see a strain that's not producing any disease. So I repeated this to about 1169 mutant strains. After actually doing this droplet infection, I also decided to just to make sure, these strains are indeed avirulent or hypovirulent, to spray onto these leaves, and this actually confirm whether these are truly avirulent or was there some inoculation effect on them. And after spray, as you can see, some of the mutants so that have show their indeed virulence. So this part of the screening, also I wanted to select one isolate that I can further study the biology of the pathogen. So I can only study biology if it's virulent. So my main aspect of looking at the biology was, how does Cercospora beticola spore germinate on the leaf? Does it show any stomatal tropism? Is whether there is any aprosorium formation during infection, and what actually happens during the infection process. On the right, you can actually see a figure, and this is way after the infection of the plant or the leaf surface with one of the strains, and you can see a lot of these Cercospora are erupting through the stomata, and that's why you can see these patches of fungal fluorescing GFPs, because they are erupting out through the stomata. So what I did was I sprayed the leaf, with a spore suspension, and incubated the plant in really high humidity, and I sampled a leaf at day 1, day 2, up to a week. So you can see in day 1 is able to visualize the spore on the leaf surface. You can see the stomata. This is done through confocal fluorescent microscopy. So by day 2 and day 3, you can see the spores beginning to germinate and kind of growing on the surface of the leaf. By day 3, the fungus is already producing this extensive network of hyphae on the leaf surface. I was not able to see any stomatal tropism. The fungus is just growing or having this network of hyphae. And by day 4, I was able to see that the fungus was penetrating the stomata, that you can see where I have pointed with the red arrow. And then also there is this function is actually producing an aprosorium-like structure. That is, it is not just entering straight into the stomata, but it appears that it is going into the stomata and forming some form of structure. I still have not dissected the leaf to try and look at the structure, so it's an ongoing process. So looking at this infection biology, had two main reasons. Was to look at how Cercospora beticola in general infects table beet. So it took about 2 to 3 days of spore germination and hyphal expansion, and we did not see any evidence of stomatal tropism. They entered the stomata within 4 to 6 days, and like they were forming acrosorium-like structures. But like I said, the second reason was also, so I screened all these mutants that were, and then I identified some avirulent mutants in my screen. I want to know, why are these mutants avirulent? So this screen will sort of create like a base to understand if some of these avirulent mutants, can they still grow on the leaf surface? Can they still enter the stomata and then not be avirulent, or is there somehow avirulent because they cannot grow on the leaf surface at all? So like I said, in my screen I did on Cercospora beticola, I identified, or I screened about 1169 mutants. And of these 1169 mutants, majority of the mutants were virulent. That is 1148 strains, which is typical of a genetic screen, is you don't identify too many strains that are actually virulent or avirulent. So out of these 1169 strains, 7 strains were avirulent and 14 strains had reduced virulence. So far, this is qualitative, or it's only visual. I have not quantified how less of a virulence they are. It's still an ongoing project, and I was unable to work on this project due to the pandemic. So hopefully once we get to work in the lab, I will again, go back and further screen these mutants. In Cercospora beticola, I was actually able to see a lot of morphological differences in my strains. So in the center is actually the wild type strain that I used to transform, and it's surrounded by strains that are morphologically different than the wild type. You can also see one strain at the bottom that's growing very, very slowly. So I think there is the mutation in this strain is not just hampered growth and development and hampered avirulence. Apart from looking at avirulence, I've also been trying to look at cercosporin production in some of these strains. However, as you can see on the leftmost, I have the TB14085, and that's the wild type strain. And that is the tricky part, is visually I cannot detect cercosporin production in this isolate. So I'm not sure, like the previous slide that I showed of cercosporin, that was in Cercospora zeamatis, it was so bright, pink. I could see with my eyes that there was cercosporin production. But with this strain of Cercospora beticola, I don't know, it may be so less that I cannot actually see. But then in a lot of my strains, I was able to look at cercosporin production, and these strains were producing higher amount than the wild type. So I looked at both light and dark conditions on PDA and V8. As you can see, the wild type, the TB14085, there is no cercosporin production. However, in a lot of these strains, I was able to identify higher amount of cercosporin being produced. So this project was then abruptly ended due to this COVID-19 pandemic, and I haven't been able to revisit this project in more than 2 months. However, with the lab opening soon, I think I'll be able to get back and then rework on these isolates. So that brings to the end of my talk, and hopefully I've been able to introduce you to the Cercospora species which pose significant threat to different crops, and how they're developing fungicide resistance, and hence we need to devise alternate management strategies. Cercospora beticola and Cercospora zeamatis, which I have worked extensively on, are suitable to understand virulence and secondary metabolism in Cercospora species, and hopefully the work that I have done in these two can be broadly expanded to a lot of Cercospora systems in other crops. So since I've done so much forward genetic screens, and I think they are effective strategies to dissect gene regulation and function, and I feel that next generation sequencing tools can accelerate genetic studies in filamentous fungi and Cercospora species. Now, going into future directions, we still have a lot of work to do on the mutant strains that I've been working in Cercospora beticola, is to identify the mutation associated with A virulence. So I've been trying to work with GeneWiz and other companies on kind of develop this target enrichment sequencing in the Illumina platform, because I've previously worked on ion torrent, and also, like I said before, do confocal fluorescent microscopy on some of these avirulent mutants to see what is the cause underlying these avirulence. Like I said, whether they are able to grow on the leaf surface or not, or they are able to penetrate through the stomata from aprosorium or not. I think it'll be an interesting aspect to look at the end, and identify the genes associated with the avirulence and characterize some of the genes and utilize RNAi-based methods in Cercospora beticola to eventually control the disease in field-based scenarios. For this work, I would like to acknowledge my previous Ph.D. supervisor, Dr. Bert Bloom from University of Arkansas, and some of the people that helped me a lot in my course of Ph.D., especially Dr. John Reidnauer and Dr. Bram Dillon, who inspired me a lot on the project and helped me in different aspects of the project. Also, I would like to thank Alex, Samantha, Hazel, and Wayne, who helped me in different capacity to complete the project in Cercospora zeomatis. I'm very thankful to Sarah and Frank, who are the PI and co-PI currently. They have, given me total freedom to work on this project in Cercospora beticola. Christopher Rivera, he was an undergraduate researcher last summer, helping me out with the transformations and screening a lot of these mutants. I get a lot of technical help from Audrey, Sean, Carol, and Kelly. I would like to thank NSF and USDA, who have been a big part in funding a lot of work that I've done through my PhD and my postdoc. University of Arkansas, Research and Extension, thanking you during my PhD. And currently I work in Cornell AgriTech. So I would like to thank them too for giving me this wonderful opportunity to work. And yes, this is most of the current lab members who have helped a great deal in this project. With that, yes, I would like to thank GeneWiz again for giving me this opportunity, and I'll take any questions now.
Great. Thank you for that insightful presentation. We did have some questions come in, so let's jump right into those. First question we received: are there any secondary metabolites produced by Cercospora species besides cercosporin?
They are. I actually did not mention, so the species that I'm currently working on, Cercospora beticola, also produces beticolin, which is another virulence factor. But beside beticolin, Cercospora, they produce flachrome is another secondary metabolite that's produced. And although we know they have different clusters for producing these secondary metabolites, but how are these involved in pathogenicity or causing disease is still not known.
Could you give more insight into stomatal tropism in Cercospora species?
So that's a very good question. So in species like Cercospora zeomatis, there is evidence of a stomatal tropism. So in this, what happens is when the fungus spore lands on the leaf and begins to grow, it is somehow able to sense or know where the stomata of the plant is. And when it's growing, it's able to reorient its hyphae towards that stomata. So although it's seen in Cercospora zeomatis, I don't have evidence of stomatal tropism in Cercospora beticola. In Cercospora beticola, I think the fungus just casts its net over the leaf, And as soon as it somehow finds the stomata, and then it is able to infect the plant. So it's really interesting in Cercospora zeomatis, but no such evidence in reticola.
Next question, what is the cause of asymmetric insertion?
So that's a very good question. I'm still not sure what are the main causes of asymmetric insertion, during these transformation events. But what I think is during these transformation events, there is a big, chromosomal rearrangement happening in fungus, and so this probably causes the cassette, the insertional cassette, to break off at some point. And I think this is the main cause of asymmetric insertions.
Will designing probes across the entire insertion cassette be a better strategy to obtain symmetric insertions?
Well, we've not tried designing these biotinylated probes across the whole of the mutagenesis cassette. I mean, it's definitely one way to know. However, I think designing probes just at the border is sufficient because designing all across the cassette will only take up the depth during sequencing, and I highly doubt it will help in any way to know whether the insertion is indeed symmetric or asymmetric.
How many strains could be sequenced simultaneously using this method?
So in my sequencing, when I was using the Ion Torrent platform, I was able to simultaneously sequence 32 mutant strains in one chip. I think, an Ion Torrent, the maximum sequencing capacity was only 2 gigs. Illumina, where you can have minimum of like 15 or 16 gigs, I think the sequencing is only limited by how many barcodes you have. I think as long as you can barcode individual genome with, like hundreds, you can theoretically, this is just theoretically, sequence hundreds of strains simultaneously in one lane.
Are 1100 mutant strains sufficient for genetic screen for phenotype like virulence?
To be honest, it is not. I think in order to have a bigger coverage in the genome, it's good to get more depth. Frankly speaking, I would love to screen thousands of strains of virulence, and that'll give even wider coverage of the genome. But depending on the resources that I had in the lab and the time, I was able to do only 1169 or so. But I would be really interested in expanding this to maybe 10 or 15,000 mutant strains, and then screen each for virulence.
Looks like in the interest of time, we'll have to stop here for today. Once again, I'd like to thank Dr. Sandeep Sharma Khatiwada for his presentation and all the attendees for joining us. Your feedback is important to us, so please be on the lookout for a brief 2-question survey that will be popping up shortly. If you'd like to watch this presentation again or any of the other webinars that are a part of GENOMS Week, please visit www.genomsweek.com. Thanks for watching.